This is work we did from 2006 to 2009, but I think it gives you a good introduction into the world of emerging contaminants. John Jackson this morning talked about this a little bit, and he said about emerging contaminants, they're really not emerging. They're something that's been around for a long time, and that's why through this process we've changed what we've even called them. And now the term we use is contaminants of emerging concern, because what's emerging is not the contaminants themselves, but our knowledge of them, our ability to measure them. So it's more the concern over them that's emerging than the actual contaminants. Many of them might have been there for many years, we just haven't been able to detect them. So we did two major efforts back starting in 2006. And this is when this was kind of a newly developing, it's rapidly developing, but this was pretty new to us at the time. USGS did some chemical work or research group in the late 90s, early 2000s, and they started to develop techniques to find pharmaceuticals, hormones, other organic compounds, but they're finding them at very small concentrations. Throughout this, we're going to be talking about things in parts per trillion, nanograms per liter, and even sometimes there we're talking less than 1 nanogram per liter. So we're talking really small concentrations. Both of these were funded through Pennsylvania DEP and the USGS. And the goal of it was, at the time, was these studies were coming out and they had sites nationwide and they were having what they found and the concentrations they found. So we wanted to look at what we were finding in Pennsylvania compared to what they were finding nationwide. These things were of importance because at these low levels, some of these things are endocrine disruptors and they can have a big impact on everything from macroinvertebrates to fish to amphibians, anything that's coming in contact with them. And they can mimic natural hormones sometimes, and it can throw off the whole life cycle of the organisms. Fish health issues have been one that have been in the news a lot and have been at least associated with emerging contaminants, but they're not very often not a direct link to one contaminant causing the fish issues. In the Potomac and even in the Susquehanna, they found some intersex fish, and it's a possibility that it's related to some of these endocrine disruptors. Sources, especially when we're getting started, there were logical sources of where we thought these things were coming from. The human sources, wastewater treatment plants were the obvious source of where these might be coming from, and you think about a wastewater treatment plant, it is a place where all these things are concentrated into a single discharge into the stream. So it's a point where these products going down toilets and drains all across an area are concentrated into one discharge. And most of the sewage treatment plants were not designed to remove organic compounds. You have a question there?
Yeah, the microbes then, you're saying either are breaking some of it down, but there's so much of it that some of it's getting through, or they can't break it down?
Some can't break it down, some there might be too much. Most of the wastewater treatment plants were designed to remove nutrients. They were designed to remove those kind of products. Now, through that process, a lot of these compounds are reduced or eliminated, but not all of them. And you'll see that there's some compounds that are almost completely removed by the wastewater treatment and some that are not. And it depends on what kind of wastewater treatment you have. Some are more effective than others. So another possible source, the major one that we were looking at, were animal sources. So antibiotics, hormones, things given to animals in manure, applied on fields, they could possibly make their way into the stream. So they were some of our first two ways we wanted to get into looking to see what we could find. Sampling for these can be very difficult. We're talking parts per trillion. You have to be very, very careful not to contaminate the samples. We've used all Teflon equipment, cleaned with methanol. Two-person, three-person field crews. Very labor-intensive. We have to be very careful because a little bit of contamination is going to be even more than you're finding in the stream. So this took a lot of effort. We did a lot of QA sampling also to try to make sure that the results we were getting were something we could understand. We did 33 blanks and we found only 4 detections. We did duplicates and spikes, and 30% relative difference is actually a lot.
What do you mean by a blank?
Okay, a blank would be a sample of like laboratory clean water.
Oh, okay.
And there's different kind of blanks we'd send in. Some we would send in, most of them were equipment blanks. So we'd clean our equipment the normal way, put the blank water through all of our equipment into the sampling bottles and sent it to the lab. Hopefully what we get from the lab is there's nothing in it, showing that there was nothing in our sampling method and equipment that would come out. Occasionally you do get some things in the blank, and it can be either contamination in what we're doing in the field, or it can be lab contamination, or it could be kind of false positives from the lab. But you hope not to see a lot of it. And what we saw, we only have 4 substances that were detected once in 33 blanks, so not too bad. Duplicates and spikes, duplicates are where we take the sample twice pretty much, send it to the lab and hope to get pretty much the same result. 30% for some samples would be very high. For some of these samples, it's not too bad because the concentrations are so low. So if we're getting 0.006 nanograms per liter in one sample and 0.012 in another, that's 100% difference, but the two numbers aren't that far apart. Since we're dealing with very small concentrations, those duplicates and spikes aren't too bad. What a spike is we collect a regular environmental sample and we split it into 2 pieces, okay? So they should be the same. And to one of them we add a known amount of compounds. Then send it to the lab and hopefully they can recover and report exactly what we put in there plus what was ever in the natural sample. So it's a way to see the recovery coming back from the laboratory. So these are just tests to see what kind of results we're getting. And these are a little bit higher variability than we would like to see in normal samples, but for some of these samples and for the techniques at the time, these were considered acceptable. A lot of times what I'm going to talk about in this talk is not what concentration we found, but whether we detected the compound or not. Because we're working at these levels, even if we saw 0.008 and it went up to 0.012 over several samples, we're kind of considering it's all kind of in the same range of results, that we were able to detect it at this kind of level. New methods were being developed then and continue to be developed constantly, and there's been a huge increase in the number of compounds we can detect and how low the detection limit has gone. Again, this slide is from like around 2012, and the last 5 years, the number of compounds has exploded greatly that the labs can analyze. We were fairly limited in this study to what was available, and it was a fairly short set of compounds. And as you can imagine, the potential for the different number of compounds out there is huge. It's thousands and thousands of different compounds possible. And often when we're doing analysis, we're looking at a very small sample of what the lab can see and what they can analyze comfortably. There's many more compounds out there that could be there, but we can't see them. General results, what we found was most of the compounds were not detected. If they were detected infrequently. The concentrations were very low. Some of the main things that influenced where we found more compounds were the number of sources, and most of the sources we ended up that were important were discharges. Mostly wastewater discharges come out as the major source of where these things is. And again, to us, that wasn't a big surprise. That's what we thought the case was. And you can imagine why. That's where most of the products that are used end up in the wastewater system. Concentration at the source and distance and flow had a big thing to do with it. The more flow you had, the more dilution there was, and the lower the concentrations were. And this was true then, and it's true now. There's still some unknown effect of what these low level of this mix of organic compounds, how it affects humans and how it affects wildlife. It's not completely understood. It's not like there's some where if you have a certain level of this arsenic, it's going to cause an effect over this. These things are at very low levels, and there's no aquatic life standards for them. There's no really regulatory standards for them. This is just kind of a list of some of the compounds that we did detect. Caffeine, interestingly enough, that showed up to be one of the most commonly ones we found. You can imagine there's a lot of caffeine goes down the drains, and it gets out through the... So that one did surprise me. Carbamazepine was one that I hadn't heard of when we started this, and it's not something we think a lot of. It's an epileptic drug, but apparently that is a drug that the water treatment plants, it doesn't seem to work well. It does not remove them. It's a very stable compound, and it moves through undisturbed. So you get things that you're acetaminophen, caffeine, that, yeah, okay, I can understand that. Some other things that you might not expect. A lot of it has to do with the chemistry. Some compounds might be very prevalent, but they break down easily. Others might be fairly rare, but they're very stable and they don't break down. So it's a mix of things, but if you notice, most of these are human use ones. We didn't find a lot of the veterinary drugs and hormones that we looked for. Phase 1 sampling, so we started looking at surface water sites, upstream and downstream of ag areas, and upstream and downstream wastewater treatment plants. We picked fairly small streams, so we looked at like combined very intense agriculture areas, and we tried to sample upstream and downstream to see if we could see differences. We looked at groundwater wells in ag areas. We did quarterly sampling for a year, and it was done at base flow, so this wasn't at storm conditions, this was at normal flow conditions. The sites were in southeastern PA, south central PA, and we looked at pharmaceuticals and antibiotics. We didn't have hormones available to us in 2006.
What frequency flood were you using for the base flood?
We were using, we have gauges in the area, and the flow had to be at or below median flow for that time, so it was no stormwater involved.
I don't know if you're looking like frequency like. Okay.
No,
Gotcha.
we're just, the idea was that we weren't getting any rainwater, any runoff into the
Right.
samples. It's at low conditions. So the samples were located across, excuse me for going a little quick here, but in an hour we got 2 presentations, so I got to get going a little bit. South Central PA, a lot of agriculture areas, small sewage treatment plants. Results from phase 1, again, this was just 1 year quarterly, so we only had like 4 samples at a site, but it got us going of what we might find. And what we were finding from, these are at wastewater treatment plants, that downstream from the wastewater treatment plant, we were finding a lot more. And you notice this says number of detections. This isn't concentrations. This is just how many results we got back from the lab that identified that they had a verifiable concentration. So you can see that downstream from the sewage treatment plants, we were getting a lot more detections. This does get into concentration. I just wanted to show that at some upstream and downstream of sewage treatment plants, the concentrations were relatively low. At others, they were very high. So this is an indication of 2 things. One, that the sewage treatment plants might have different capabilities of removing. And probably the biggest thing is that the percent of flow that's going into the stream from the sewage, so how much dilution is happening. If you have like in Killager Creek, half of the flow, 49% of the flow is coming from the sewage treatment plant. So there's not a lot of dilution, natural dilution in the stream, and we have a lot of detections. In Spring Creek, only 7% of the flow was coming from the sewage treatment plant. So there was a lot of dilution in the natural stream, and the number of detections was much lower. So the sewage treatment plant is a concentrated source. Once it gets in the stream, it dilutes. The more dilution you have, the less compounds we could find. Again, this wasn't shocking, but it was good to actually be able to see it and measure it. Same situation from a different situation. These were the animal feeding operations. So these were the intense farming. And what we see is upstream and downstream, you see some slight differences. One, the number of detections is very low. And this isn't as clear cut that downstream of the feeding operation is significantly different than upstream of the feeding operation. Now some of this is that we were looking upstream and downstream of a big feeding operation, but there might have been more upstream. So the flow coming in wasn't altered greatly. We're not finding like a smoking gun below some of these CFOs. We found maybe a few more of certain compounds and a few less of others, but most of the ones we found were human use products, not animal products. So we weren't very convinced that the animal operations were adding a lot to the concentrations we were seeing. So the phase 1 we got through in a year. Few detections, more detections downstream from the wastewater treatment plants. Everything was very low concentrations. Very few detection in the ag streams, and the stock wells, we got almost nothing. There was almost no detections. So again, through that, it kind of focused us toward more where the sewage treatment plants were probably our biggest thing to concentrate on. So we focused more on sewage treatment plants, added hormone and organic wastewater compounds, and we looked at sediment also. And when we looked at hormones, this is a pattern we saw often. There was 1 or 2 or 3 compounds that we found regularly. Most of the other ones we found almost not at all or infrequently. So these 2, this is a female and a male hormone, that we found somewhere between 50 and 40% in the water sample, but we found a lot more in the sediments. So when we looked at the sediments, some compounds are going to be more likely to gather in the sediments depending on their structure. They're going to bind to them. Other ones might be found more in the water column. Again, very low concentrations. Organic wastewater compounds are just a group of organic compounds that are typically found in wastewaters. It's a very wide range of compounds. It can be everything from industrial type compounds to natural compounds, but they're compounds that are typically found in wastewater. Similar pattern where most of them we didn't find. So these ones we would send to the lab, say for 50 analysis of 50 things, 48 of them would come back as nothing found, 1 or 2. So most of these were coming back as nothing. Interestingly, some were found in sediment more than water, some more in water than sediment. But again, that's due to their chemistry and their interactions. Polycyclic aromatic compounds were found more in the sediments. And one thing we didn't, at the time, I didn't think of much, the flame retardants and plasticizers were mostly in the water samples, not the sediments. And a lot of these, this is interesting, come from clothing. A lot of babies' clothing, a lot of products are put in with these flame retardants. And when you wash them, they come out to the wash water and out to the sewage treatment plant. And these are ones particularly that don't seem to come out in the wastewater treatment. So they end up into the stream right away. Again, detection frequency. This is stream water in blue and sediment in red. And what I put this up mainly for is you can see that in the antibiotics and hormones, you can see some are more in the sediment, some more in the water. When you get to the wastewater compounds, the organic ones, most of them are in the sediment, so they concentrate into the sediments. But in doing that, in some certain circumstances, they can remobilize out of the sediments into the water, and they might be available to the invertebrates, the fish. So being in the sediment might actually make them more accessible to certain organisms at certain times.
Might be able to state just briefly, how did you sample the water, and how did you sample the sediment, and then what was your analysis technique for the sediment in the water? Is that something you said briefly?
I could talk about the collection more than the analysis, because the analysis were done in our lab in Denver, Colorado.
Okay.
So they have their standard methods and all. The stream water was collected either by, if it was a small stream, we would weight it. We pretty much divide the stream into sections, and we take a equal width collection, which means, say we break it into 5 sections, we would collect a volume of water from each of those 5 sections into a Teflon bottle, composite into a Teflon churn, mix it up in there, and then distribute it into the bottles to be shipped to the lab. Sediment was collected in depositional areas using stainless steel scoops and bowls and a sieve. So we would scoop out a certain amount of bottom sediment, looking for the most organically rich sediment we could find, wash it through the sieve, collect it into glass jars, and then that was shipped off to the lab where they analyzed it. So anyway,
I'm sorry.
No, go ahead.
While we were still on that, I was going to ask how you guys calculated the flow percentage.
for most of there's 2 things. You can actually take a discharge measurement, and then you can break it up. For these, what we did, that's an equal discharge. What we did was an equal width, so we broke it up into, say, 5, 7, 9 sections, and took an equal volume for each section. So it wasn't necessarily flow weighted, but it was equally distributed across the stream. Again, phase 2, hormones and wastewaters had a similar pattern as what we saw earlier. We had a couple compounds that were detected frequently, although at low concentrations. Most of the compounds were detected rarely. Some compounds were more in sediment, others were more in water. And that's just estrone, DEET, which is in bug repellents. That showed up a lot in the sediment samples. The final phase of this was looking at, rather than where it was coming from, where it might be going and what impacts it might be having. So through DEP, we've been sampling with them the Water Quality Network. So we have a network of sites across Pennsylvania that we've been sampling, and DEP's been sampling in the 50s, I think.
Some of them go back to the early 1950s.
Yeah. So we have this wide network of sites where we have historic basic chemistry data. So we took a subsample of those sites, 27 of them, that were located close to a drinking water intake. And the idea being is not just what's in the stream, but what might be available to get into the drinking water process. Now again, the drinking water process also removes a lot of these, so it's not, what we find here isn't what's going out to the tap, it's just what was available to the drinking water intake.
Did you test what was coming out of the tap?
That has been done before, and the drinking water process removes most of it, but there are, again, a couple compounds that can slip through. But what's coming, the numbers you see here would be greatly reduced to what's going out into the drinking water supply. So I don't want people to think this is the drinking water supply, this is just the water in the stream that's accessible to. So we had 27 sites near drinking water. We looked for pharmaceuticals, antibiotics, and hormones. So this is the sites from all of the things. So you can see we had it kind of statewide. There was a lot central in south central PA because that's where the initial studies happened, and that's where there's a lot of good farmland and area for us to work in. But we also looked out in Ohio and in the Delaware. I'm not going to talk about it, especially for time here, but we also, through USGS in Leetown, West Virginia, did some fish health surveys where they collected fish at these sites and looked for the intersects, they looked for health, they looked at contaminants in the livers and things. So that's a whole other project, which we won't be talking about today. These samples we did collect at various flows. So we didn't target just base flows, or we didn't target stern flows. We tried to get a mixture of flows. So we had some high, some low. They were collected with regular WQN, so we have background chemistry with all of them. There was a separate project looking at E. coli, Giardia, Cryptosporidium. And these streams varied from very small streams, maybe 10 feet wide, to the Delaware River, to the Allegheny River, so the Susquehanna, Harrisburg. So there was some big and small streams. At all 27 sites, we found at least one compound. So there was no sites that were completely clean. 80% of the pharmaceutical compounds were detected at least once, 50% of the antibiotics, and 40% of the hormones. But notice, at least once. Most of them were not detected very often. The common pharmaceuticals, and I know caffeine's not really a pharmaceutical, but it was in our pharmaceutical schedule at the lab. But you can see there's common things, caffeine, acetaminophen, Benadryl, so things that you would be expecting heavy use, and some things that aren't, the carbamazepine. But that was very similar to the studies we had done the year before and the year before that, and it was very similar to the studies that USGS had done nationally. So these were things we were finding that were also being found nationally, so this wasn't a specific indication of problems in Pennsylvania waters, this was more of a general condition across the United States. This is just percent detections. For every sample we sent in, about 78% of them came back with caffeine hits. But you can see this pattern where you have a couple compounds that are detected commonly, and the other ones fall off quickly. So these ones we were only finding a couple times in all of our samples. Antibiotics, the biggest thing here is they're all human use ones we found. We were not finding like the agricultural, the veterinary type ones. They were all human use. Which again leads us more to, it's probably more of a wastewater discharge thing and not a farm runoff situation. Again, antibiotics, one that we found a lot that we didn't find at all. And a lot of this just has to do with the chemistry of the compound and how it interacts with wastewater treatment and how it interacts in the stream. Some of these indicate that they're just very stable compounds and they do not break down easily. Another problem we had with this study at the time is that some of these compounds will break down in metabolites quickly. And if we're looking for the parent compound and it's broken down, there might be remnants of this still there, but we're not seeing it. So because some of these weren't found, it doesn't mean that when it goes in, it completely disappears. It just means it's either being broken down or it's being changed into something else that we're not measuring. So there's a lot going on here. It's not a very simple situation. Again, a couple commonly detected hormones, and some of these might be in pharmaceuticals, but some of these are natural too. We all put out hormones, they all go down the toilet, and they all end up at the wastewater treatment plant, so there's a chance for them to go out. So this isn't just pills being dumped down the drain. This is some natural processes going on also. Again, a couple hormones that we found commonly, but it drops off quickly. So if we had a list of 20 hormones we were looking for, once you get past one or two, you rarely found them. So the big take home is that most compounds we were not detecting. However, there were a couple that we did find pretty consistently. We also looked at some reference sites to try to get away from point discharges. And what we found there, the 5 reference sites, we found a lot less detections. At our other 21 sites, we found a lot more detections, and the concentrations were much lower. So that was a good sign. But at some of these, there's very few discharges, and we were still finding some compounds. Some of the big patterns we start to see and where we would see it is the hydrologic conditions. So a lot of times people think of you get heavy flow, and that's going to wash contaminants into the stream. In these cases, since the contaminants were coming from the wastewater treatment, flow would actually dilute what we were finding. So at high flows, a lot of these compounds would come back as non-detects. Where we found them the most was at the summer low flows. So when the stream is concentrated, that flow from the wastewater treatment plant is now making up a bigger percentage of the flow, and it's not getting diluted, and then we'd be able to find these things more easily. Number of discharges. This wasn't a very sensitive measure. We weren't looking at like how much wastewater was coming in. We just did a simple look at how many permanent discharges there were in the basin, and we found that the more permanent discharges there were per drainage area, the more compounds we found, and that's not surprising either. In land use, the same thing. The more forested there was, the less we found. So again, highest concentration was during low flow. There were a few times when that didn't work, and in those cases where if we're seeing a compound higher at high flows, that means it's probably getting washed in over the land versus from a sewage treatment plant. This I just want to show you. One, we had a lot more, the drinking water intake sites we did for 3 years quarterly, so we had a lot more samples than the other sites. But you can look at the percentages here. Downstream from the wastewater treatment, pretty high percentage. Downstream, or near the drinking water intake, much lower. So dilution and breakdown and changing is happening because fortunately we don't have too many drinking water intakes that are very close to wastewater. Hopefully not, anyway. Again, this gets what the number of discharges in the thing. It was interesting. If we work it into a low, medium, and high category, and at the low and the medium, and this again, it was the number of permanent discharges kind of divided by the drainage area. They were about the same, but once you got about 0.28 discharges per drainage per square area, it shot way up. So it seems like if you have a few discharges, you're okay, but once it gets past a certain point, then it starts to build up. So, and again, this isn't surprising. The more discharges you have, the higher it goes. But it seems like there's a threshold there that shot it up. In land use, it's kind of the opposite. The more forested area you had, probably less discharges, the less inputs. We did a little bit, we looked at seasonality for some of these to see if you could see, do people take more Benadryl in the spring, and does it come out in samples? So we looked at this at most of the compounds, and most of the answers were no, it doesn't. But these two it did, and acetaminophen, which is a painkiller, seemed to peak in the winter. So it seemed like it's possible more of that product was being used, going through the wastewater treatment and out to the stream, where sulfamethoxyl, which is a antibiotic, seemed to have a pattern where more was being used in the fall. So I don't know if I'm going to explain that other than that's what we found. But there seemed to be a seasonal difference to those. But most of the other compounds were not seasonal. We didn't see a change in them. And the hormones, again, you wouldn't expect that because that's more something that's happening all the time, where in pharmaceuticals, people might be taking more of one product in a certain season than another. So overall, few compounds detected rarely, low concentrations, downstream wastewater treatment plants were the most concentrated areas, and at low flow. So if you have a heavy discharge into a small stream in the low flow summertime, that's when the most concentrated times can happen. But that's important to know if you want to relate it to fish health and to other things, you have that information. Number of discharges and land use. So again, the more discharges you have, the less forested area, the more likely you are to find them. Not surprising. And as of now, we're getting much, much better at detecting these and knowing where to find them, but we still don't know much of what it means. So we don't know what a concentration of, 0.12 of something, what that does in the environment. And probably the scarier thing is, what we don't know is that if you have 100 compounds out there at this low level, mixing together, what the effects of that are. And that's going to be very difficult to tease out. We have a report out on this. If you go to our website, you can download it. If you go to, probably the best place to go is the pa.water.usgs.gov and look under reports, and you can find that, plus lots of other reports.
Hold on 1 second.
It's pa.water.usgs.gov.
Thank you.
Okay, and there's a publications tab under there, and you should be able to find it there. I want to give Gary some time, so there's some other work that was done, but you can find it on the USGS websites also.
Thanks, Drew. Following Drew's lead-in, I'm Gary Walters. I'm with the Department of Environmental Protection, the Bureau of Clean Water, and through reorganization, the name of the division is now the Water Quality Division, not the Division of Water Quality. I am the chief of the assessment section of water quality, and the Division of Water Quality, just so you have an idea of how the department is organized, is responsible for developing the water quality standards, or the criteria that all surface waters of the Commonwealth have to meet, or all discharges would have to meet in order to protect our designated uses of streams. My particular section, my staff and I, we are responsible for developing methodologies to evaluate all the data that's collected from the stream, determine whether or not we are meeting the criteria, or do we have protected uses, and are they being supported. We also do a number of other things as well. Myself and my staff are all biologists, and we do some of the monitoring as well. And one of the big projects that we had started in 2012, and this is the Susquehanna River study. It's sort of an offshoot of this project that Drew worked with other people in our bureau for the 2006 to 2009 study. It's a little bit related, but it only simply because we learned a little bit from that project before we took this one on. So what I'm presenting to you is largely the work of my staff, Amy Williams. She is the one who developed the entire sampling program that we were going to undertake. We tested for a total of 274 analytes of these types of things. Passive samplers, and I'll talk a little bit more about this. Just so you know, this is a passive sampling device there, at least the container that it's in. We looked for total estrogenicity, pharmaceuticals, a variety of wastewater compounds, pesticides, both the organochlorine, which is the old class of pesticides, and those that are in current use, the PCBs, the PBDEs, which are the flame retardants, and the PAHs, or the polyaromatic hydrocarbons, a petroleum-based product, TARs. We did water grab samples. Water grab sample is you just wait out in the stream, and many of you have done volunteer monitoring. You've done the water grab sample. You wait out, you get a bottle of water, and it goes off to the lab. We've done some of the QA things that Drew has talked about. For that, we also did total estrogenicity, pesticides, triazine pesticides. Triazines are herbicides used in no-till farming. The most common one is atrazine, and glyphosate, which is known as Roundup. For sediment, we looked at the pesticides, both the old organochlorine and the current use types, various wastewater compounds, hormones, and metals. When we did the sediment sampling, we did a little bit different than what Drew talked about. Because we were looking at metals, we did not use any metal containers. All of our containers were plastic to make sure that we weren't contaminating our sample with any metals off of the metal tools. For the passive samplers, like I said, I'll talk about. These are two of the passive sampling devices. The Polar Organic Chemical Integrated Sampler, which is this device here. These are membranes that concentrate organic compounds. The semi-permeable membrane device, most of us would know it as a dialysis bag, was the other type that we used. And again, these are to collect the compounds from the water. Drew had talked about the fact that these compounds are present at very low concentrations. We were detecting things at the nanogram level. So he talked about parts per billion, parts per trillion, and we went to parts quadrillion. And in order to detect many of these things, we have to deploy a device like this. These devices went out for a total of 30 days. I forgot to mention, I do have 25 handouts of the PowerPoint up here if you want to pick one up as you go out of the room. It does have the contact information. We sampled fall of 2013, spring and fall of 2014, same again in 15, 16, and we will be doing the same this year. The number of sites varied. In this year, we're planning on 27 sites, and we're going to expand farther out. The reason we focused on the Susquehanna River, there had been a long-term trend of smallmouth bass declining in the Susquehanna River. And in 2012, nothing was getting better, and we had to start a project to determine what was going on. And part of that was to determine what was out there that might be one of the problems. And these contaminants of emerging concern, of course, came onto the radar. We know they're present based off of the study from 2006 to 2009, so we decided we better look a little more closely. So passive samplers were part of that. Then the sediment, the same thing, fall and spring sampling beginning in the fall of 2013. We haven't increased sites for that. We have gotten results in the sediment. We don't really know what to do with the results. We don't have standards to tell us whether or not we've got a problem. We're not finding things at high enough levels that suggest that there actually is a problem. There's certain compounds that concentrate in the soils versus others, but we're not seeing anything at this point that would be alarming. So we're not expanding that too much just because it's very expensive analysis and we don't know exactly what we're going to do with the data. Yes?
How long were the passive samplers in Susquehanna?
I think I have a slide about that, but jumping ahead, in case I don't, they're deployed for a total of 30 days. And then the water grab samples, total estrogenicity, current use pesticides, triazines, glyphosate, and in this year we're looking at about 54 sites for the total estrogenicity. Pesticides, triazines, and glyphosate as necessary. We got most of our data from the passive samplers. We had a number of labs where this went to. The old class, the organochlorine pesticides, they went to DEP's Bureau of Laboratories for analysis because our lab was able to do that. Most of the current use pesticides analysis was done at one of the USGS labs. We have a contract through Drew. I'm not sure where all.
Yeah.
We go to a number of different USGS labs for this analysis. The hormones went to USGS lab. Many of the pharmaceuticals.
It's confusing because some went to our lab in Denver. We have
Right.
research labs. So a lot of this is still in the research phase. It was a research lab in Columbia, Missouri that did a lot of it,
Right.
and some of it was shipped out to other research labs. So
It went all over the place. In total, over the last 5 years, we've spent close to $1 million on analysis just for these contaminants of emerging concern. And then sediment sampling went all to the DEP Bureau of Laboratories. This is really a little bit out of order, but what we found was no different than what was found in the 2006 to 2009 study. We are detecting some of these compounds everywhere. None of them occur everywhere, and it varies. Some places appear to have a higher level than others. One of the big keys to this, and Drew talked a little bit about some of the things that they were seeing, certain compounds are not going to be there at certain times of the year. It's all based on when they might get used, like the atrazines or the triazine compounds. Because they're used in no-till farming, they're used prior to the farm being the field being cultivated, so they will be applied. what we've discovered over the past couple of years, that the month of March seems to be the month that we need to actually sample for the atrazine or the triazine compounds in order to detect them. Otherwise, they will not be there. When they hit the water, they break down rapidly and go into their breakdown products. So you have to be there at the right time. That's part of the reason why we use the passive samplers. Many of these things are going in an episodic event. If you're not there at the right time, you won't detect them. And because they are there at such low level, we have to use those type of devices or you can't determine whether or not you detect them. We did have some controls that were outside the Susquehanna Basin in the Schuylkill River watershed, the Lehigh and the Delaware River, as well as in western Pennsylvania in the Allegheny and the Youghiogheny River. And these are our sample sites across the state for the sediment and passive sampler locations in 2012 to 2016. There'll be some minor changes for 17 with some additions. We did get results that kind of surprised us because we got results that were higher in places we didn't expect. This here is Contaminants Creek in Mercer County, and it was one of our highest hits for some of the hormones. We also got hot hits on the Delaware River at Trenton for some of the compounds that surprised us. It was much higher than other places in the state. But again, a lot of this is episodic, so we don't know if we're really seeing anything here. The big thing was there was no pattern to these results. And this is showing some of the results of the total estrogenicity graph. We can do that analysis if you're doing the total estrogens because it's taking all of the estrogen compounds that are there and do a sum of all of those. So we can do those from a water grab sample. If you're going to get the individuals, we pretty much have to do the passive sampling. As Drew pointed out, the number of what was detected was the naturally occurring hormones, a few of the synthetics. The synthetics were only detecting downstream of wastewater treatment plants, which really isn't a surprise. We concentrate all of our sewage into one point and discharge it, so you're going to concentrate those types of contaminants that way. This sample right here is the Schuylkill River at our water quality network station out here at Route 100. That was one of the ones that was non-detect for total estrogenicity. This is a one-time grab, and I think it was only in 2013. And we haven't done it since, but we're going to expand to all of our water quality network. Our water quality network is a total of 177 stations statewide, so it covers the entire state. Our biggest areas of concern are out here in the Juniata River and over here in Lancaster and the Conestoga. That's where we're detecting a lot of the hormones. This happens to be high agricultural areas. Again, it's not surprising. Estrogen is ubiquitous. Every biological being is producing it. Most of what we're seeing probably is a plant hormone versus animal. So it's everywhere. What levels are too much in the water? We don't know. That's the big problem. We really don't know at this point. We know far less than what we know about this stuff, is the sad thing. Lots of research needs to continue to occur. Pesticides, grab sampling. These are our results, and again, it's the agricultural areas that tend to be high, the highest locations, not a surprise. At least for the Susquehanna study. For the contaminants of emergent concern, the Juniata watershed had some of the highest results that we saw. Hormones were very common for the ag areas in the same way for pesticides. Wastewater indicators, they were ubiquitous, but again, they were where we had wastewater discharges. Control areas were not references because they're impacted as well. Like I said, our controls on the Youghiogheny River and the Delaware at Trenton had some of the highest results that we saw. They were not lower than what we were seeing in the Susquehanna Basin. Where we did have reference quality sites, they were good. We didn't get very many detections, which again mirrors the 2006 to 2009 study. But this is probably the key piece to this. Hard to come to any definitive conclusions.
So the last point, basically, we just don't know how toxic these are in these low quantities.
These things are not toxic. Except for like the pesticides, that their role is to kill something.
Yeah.
Most of these things are not toxic. We don't know exactly what their effect is on the animals in the
You
stream.
pointed out that it affects the hormones or picture the frog.
Not like it's not going to kill the organism outright, but it might affect its reproduction, which over time
Right.
can
Kill.
eliminate the organism from.
Right.
Yeah,
so
that's sort of like being dead.
Yeah, but it's not. It might not be killing. But it's different than if like arsenic's released in the
Right.
water, it's going to kill that fish. This might slowly reduce its reproductive rate.
Right.
And over the next 10 years, that fish might not be viable anymore.
Right.
So
That's the
there is
problem.
differences.
That's the
There is
problem.
differences, but
Yeah.
yes.
We're not saying there isn't a problem. We don't know exactly what triggers all of it. One of the studies that was on Drew's final slide was a study by Dr. Vicki Blazer on intersex condition. She's in their Leetown lab. We're actually working with Vicki on another project for the Susquehanna River to determine whether or not smallmouth bass in the Susquehanna River are immunosuppressed due to these contaminants. Because these contaminants can also, they mimic hormones, and that can have an effect on the immune system, either preventing it from reacting or overreacting or causing autoimmune type diseases. Intersex can also occur through different pathways. It doesn't have to necessarily be that hormone. Atrazine was one of the things that we thought was the big, this is what's causing the intersex. Turns out that it doesn't bind with the estrogen hormone, so it's not the big It does have estrogenic effects, but it doesn't bind with the hormone receptor, so it's doing it through a different pathway. So when you say toxicity, at least from my perspective, there's a completely different role. Toxicity equals death instantly, not something typically drawn out. These compounds affect the biology in a different way. It's that we just don't know exactly how it's doing it. We're going to continue doing the study as we have been. Like I said, we're going to expand to our water quality network. We're going to be doing more work in the Delaware Basin as a whole, the Susquehanna, or the Schuylkill, and the Lehigh, as well as the main stem Delaware. The sediment sampling for hormones and wastewater compounds, that will continue. Again, it's just going to be basically maintain the status quo because we don't know for sure at this point how we use the data, what it really means. And then passive sampling will be expanded for a number of the compounds. Of those 274 compounds that we did test for, we got detections for just a small subset of those. I can't tell you at this point how many we got detections for because we still have some outstanding data that may show some additional detections. And like I said, this work is mostly of my staff, Amy Williams, and that's her contact information. It's also on the handout I have. My contact information is gawalters@pa.gov. And my phone number is 717-783-7964. If you go to DEP's water quality website, you can find contact information for this particular division. A lot of it's going to bring you to me. So there's many ways that you can get in touch with us if you have additional questions. So with that, I'll open up to questions for both of us.
So are there efforts underway at either USGS or the state, like trying to tox test or sub-tox, if you want to say some kind of no effect level for some of these concentrations?
At least for DEP, the answer to that would be no, we're not going down the route of toxicity or what the sublethal effect would be. We are working with Vicki Blazer to see if we're seeing some manifestation of an immunosuppression in smallmouth bass, but beyond that, we're not.
The answer is yes, but it's very difficult because, again, most of these concentrations, it's not causing, you can't just put them in by the time you get to a level that's causing a definite effect, they're not levels that we're seeing in the environment. So it's not really realistic. What's more difficult is it's probably this combination of things at low levels that might be causing it, and that's very difficult to you come up with a million different combinations. So.
With that, if there's no more questions, thank you very much.